The first step is the cell procurement. This can be obtained by two different methods. The first one consists in taking a cell or tissue biopsy from the living animal, or from the post-mortem recovery of the tissue. These are called primary lines. The second method, on the other hand, involves the use of pluripotent cells. The next step involves maintaining and harvested cells in culture. The next step involves maintaining the harvested cells in culture in order to first achieve cell proliferation and then, eventually, their differentiation, with the final aim of increasing the biomass. The initial cell proliferation takes place using a small flask, in which they can grow in two-dimensional sheets, anchored to a surface. This method represents the standard for the lab cell culturing method, mainly devoted to research activities. However, for larger-scale industrial cell production, suspension cultures in bioreactors are required in order to increase the efficiency of the process. These systems require to generate a great number of cells, minimizing the resource consumption, such as the medium or the material, time, and handling step. Regardless of the final application, that could be industrial or laboratory, the current cells culturing method involves the use of specific growth media. The growth media contains all those substances that are necessary to ensure the cell proliferation, such as glucose, inorganic salts, amino acids, vitamins, growth factors, antibiotics, and antifungals. Other substances can be added to confer organoleptic properties such as proteins or pigments that give a meat-like color. However, to date, viable alternatives to classical culture media are being sought, where the goal is to introduce into the medium edible byproducts or waste products that ensure the rapid cell growth. The same principle should be applied to the fetal bovine serum, which is essential for the cell proliferation, but not at all ethical and sustainable. The tissue fabrication and maturation processes are accomplished necessarily providing cells the correct environment in which they can adhere, proliferate, and differentiate. Scaffolds are three-dimensional structures characterized by correct porosity, texture, architecture, mechanical and chemical properties, suitable for the specific cell type maturation. Moreover, considering the food engineering purposes, scaffolds must be biodegradable and/or edible, and their structure is involved also in the final organoleptic properties of the final product. The traditional approach, coming from the tissue engineering, involves the scaffold fabrication and subsequent cell seeding in a 3D culture. Scaffold can be obtained through tissue or organ decellularization, or can be biofabricated. There are several different techniques for scaffold production. The main disadvantage is the difficulty to have cell migration for thick structure and low cell density control. Recently, another approach is gaining importance: the biofabrication. This process allows to produce a cell-laden construct making use of material containing cell and biological molecules that are mixed together. The main advantage is the possibility to fabricate thicker structures and to control the spatial arrangement of cells, even of multiple types. The 3D bioprinting is a biofabrication technology based on the additive manufacturing, which allow the precise cell positioning, density deposition, precise control of the structure and ratio between different cell populations in case of multicellular constructs. Cell material mixtures used in the process are called bioinks. 3D bioprinting represents a possible game changer in the field of in vitro meat production, assuring high scalability, complexity for field process to mimic the real meat structure, minimal energy expenditure, and lower CO2 emissions. Whatever is the construct fabrication, cells inside the 3D scaffold must be placed in a bioreactor for dematuration. Mechanical, chemical, and eventually electrical stimuli are required to complete this stage through dedicated culturing systems. Bioreactors are closed automated systems in which cells can proliferate, differentiate, and maturate within the construct forming tissue. They provide a precise control over relevant variables such as temperature, oxygen concentration, pH, and cell density. Today, several types of bioreactor are used. Most common are the static culture, spinner flask, and perfusion bioreactors. Inside the bioreactors, cells proliferate and then go through differentiation. To allow an industrial scalability, products and process must be tuned to high-volume production lines.